Hi, my name is Montanna Castle, and today I will be explaining the 4 different levels of the protein structure. The first level is the primary. It looks something like this, which is a chain of amino acids of a single chain, also known as a polypeptide. This amino acid sequence is controlled by the nucleotide sequencing and the DNA coding. They have covalent bonds, which are the strongest bonds, and they do well in aqueous environments. Alright, which is right here. This bond between them is called a peptide bond, and how this occurs, well, first I'll explain what is inside each amino acid. So within each amino acid you have 4 different types of groups. You have your central carbon, and then you have the R group, and then you have the carboxyl group, which is carbon double bonded to oxygen, and then you have HO right here. And then next to it you have your amino acid identifier group, which can vary, but right now we'll use NH, and then you always have a hydrogen. And basically what happens is, so, one second. So say you have 2 of these. You have one right here, so I'm going to draw the same exact thing. CHO, double bonded to oxygen, hydrogen, your amino acid group, and your R group. And then I'm going to replicate the same thing, but it varies with each amino acid. So here we go. And what the peptide bonds do, they just hold the chain together to stay in sequence. And then here's the same thing again. And what happens here, which is called a condensation reaction, is the OH and the H combine, and then you get H2O. So you lose them, and then when it comes down to here, the N and the C from these 2 groups will connect, and this is your peptide bond. Again, this is a very strong structure. It has covalent bonds, and overall it is the most protective out of all of them. And next I will be talking about the secondary structure.